Rise and shine, it's game day, gonna give it all come what may. Fueling up with the best in mind, DHA's the secret that you
Hello and welcome to The Good Oil, a podcast series dedicated to exploring the impact role of DHA in nutrition and wellbeing. I'm your host, Bassam Hallak from New Omega Ingredients. In today's episode, we're diving deep into the science of omega-3, focusing specifically on how you can quantify the levels of omega-3 in your body to ensure you're getting the most impactful benefits. We're not just talking about why omega-3 is important. We'll be discussing how you can measure your omega-3 levels, what those numbers mean, and how you can optimize your intake to improve your health and well-being. But before we go into specifics, I'm thrilled to have some incredible experts joining me today. First up, we have Lalen Dogan, the Vice President of Fatty Acid Labs. Lalen has a wealth of knowledge and experience in omega-3 industry, spanning both scientific and practical applications of fatty acids in health. Lalen, welcome.
Thanks for having me, Bassam. It's a pleasure to be here.
And we're also joined by a returning guest who is no stranger to the good oil, Greg Peoples from Wollongong University. Greg provided us with amazing insights during the first series of the impact of DHA on sports performance. Welcome back, Greg.
Excellent. Thanks for having me again. And this is really exciting because we've got three in the room. And to be here with Lalen as part of Fatty Acid Labs and also the university, and then yourself, this is a real think tank that we've got going.
Yeah, feelings mutual.
We've got you and I on this side and Lalen's on the other side.
Okay. Bit of AFL
we're
rivalry.
balanced. Yeah.
There you go. Richmond and Carlton. It's fantastic to have you both on the show today. So let's get right into it. I know the listeners will be eagerly waiting to hear about this, the phenomenon of omega-3 levels in blood and how we can quantify that. Lalen, let's start with the basics. What exactly is the omega-3 index and why is it so important?
Great question. The omega-3 index is the percentage of EPA and DHA in blood. Essentially, the test provides a snapshot of your omega-3 status based on your diet and supplementation intake. And, we need to be mindful, omega-3 supplements is one of the widely taken supplements with the most amount of research. So it's had over 50,000 published papers as well as 5,000 human clinical trials.
So it's not just about taking the omega-3 supplements, it's about knowing how much you're actually absorbing and how it's affecting your health at a cellular level. This brings us to an important question. Do omega-3 6 to 3 ratios matter? Greg, what's your connection here and how would you elaborate on that?
Yeah, look, this is a really interesting topic of debate. And in fact, if we had a table, I could put two groups of scientists on either side of the table. And we've done this in the past, actually, at our university. We've had actually sort of a debate on this topic. Is it the ratio that's important or is it the provision of the long-chain omega-3s? It's always a really fascinating debate. It doesn't necessarily bring the sides together, but it certainly makes people think a bit more. So if we just step down a level, what we do know, they're both polyunsaturated fatty acids. So that's critical. We also know that omega-6 and omega-3 are both critical for human health. That's a really important thing. So, it's interesting that sometimes the omega-6 gets given sort of a bad name, but in reality, we know there's just as important roles physiologically for omega-6. We've just got an interesting situation that post-industrialization, we've lost the amount of omega-3, and particularly these long-chain EPA and DHA fatty acids in the diet. And the post-industrialization means that the omega-6s have kind of grown in the amount that we take in. And it's sort of not surprising looking at our Western diet overall. So the question is, do we need to actually consider the ratio or just the provision of EPA and DHA? Is that enough, despite a background omega-6 potentially being the classic Western diet, which is, essentially we all agree a little bit high potentially. We go back to animal models, and what's really interesting, and this was my PhD supervisor that actually attempted to answer this question. So he manipulated the diet of animals, and this is rodent models, for high omega-6, low omega-6, high omega-3, low omega-3, EPA, DHA. And what was really interesting, it didn't actually matter what the background was. It was actually, we needed to provide the EPA and DHA. So in other words, it's not a competition per se between them. It's just that the tissues generally in a Western diet aren't presented with enough EPA and DHA. But if you do allow them to be presented with EPA and DHA, they preferentially incorporate, because what we're talking about is the membranes here, they preferentially incorporate the EPA and DHA. We recently looked at this play out in like a pseudo human observational trial, and this is going back 2 or 3 years, where we had a college NFL team, and being in America, obviously there's a high omega-6 background that, and that's what we see in that particular population. And it was pre-season, it was 4 weeks to go, and the dietitian said, look, I've got this omega-3 supplements and algal oil. Can we give it to the players? Can we see what sort of effect that would have? Now, we didn't change any of the background diet, so we left it the same. Now, what's interesting, and we measured the omega-3 content as well as all the fatty acids through the process that Lalen's going to describe here today with the finger prick blood sample, is Western diet in America, college athletes, they were football players, pretty high omega-6s. And so if we looked at the ratio, they were pretty high. We just simply presented 1,000 milligrams of EPA plus DHA through an algal oil, and lo and behold, we could raise the index and lower what we call the arachidonic acid to EPA ratio, which is 2 specific fatty acids to represent that class of omega-6 and omega-3. So in other words, we could change the outcome measures without actually changing the omega-6 background.
Yeah.
Now, what that does raise is, should we still be attaining to lowering our omega-6s? And the answer probably is, but it's probably from a wider diet quality point of view, because many of these things are coming through processed food. So that's another story altogether. So I think the take-home message is that we're just simply not presenting enough EPA and DHA to the body's tissues. And if you do, the tissues will recognise those and incorporate them into the membrane.
Yeah. Today we're talking about omega-3 index and quantifying, the omega-3 quantities in the blood or in your membranes, as you describe it. So ideally, where do we need? And we've spoken about this in the previous podcast, but ideally, where would we need to be in terms of ratio of omega-3 to omega-6?
Yeah, and look, we can talk about it as the test goes. We'll describe the test a little bit more, but we've got a couple of different ratios. So if you look at the broad background diet, the ratio is a number of 5. So in other words, 5 omega-6 to 1 omega-3, and we're looking to reduce that ratio below 5. If we look at the 2 specific fatty acids that I talked about before, the arachidonic acid representing the omega-6 and the EPA, the eicosapentaenoic acid, representing the omega-3, we're trying to reduce that down to a level of below 11. If we go back to that NFL study, they were way above 5 and way above 11. And now what's interesting, we only had 4 weeks with them, and we were giving them 1,000 milligrams of EPA plus DHA. We didn't actually get them below that, but we're on the way to actually reducing those ratios down to those 2 numbers that I just described.
So that makes a lot of sense. So we're testing omega-3. It helps us understand whether, where we are in sort of the optimal range, in particular with, omega-3s, and that allows us then to adjust our diet or supplementation regime that we want to, each individual takes on board. Now let's talk about the benefits of testing. So Lalen, why is it important for individuals to measure the omega-3 levels?
Yeah, good question. We are all unique with differences based on gender, age, weight, diet, lifestyle, and even genetics as well. And omega-3 is an essential nutrient that our bodies can't produce on their own. So in other words, we can only obtain these nutrients through our diet and supplementation. Now, I want to emphasize that whilst we strongly advocate a healthy lifestyle and a well-balanced diet, the reality is that even if you follow the Heart Foundation's recommendations of eating 2 servings of fatty fish like salmon and tuna every week, you're still not in the desirable range of the omega-3 index. And then it's the economic factor that you need to factor in as well, right? I mean, how realistic is it for us to expect the wider population to be consuming that much fatty fish every week? And then, of course, you have these subgroups of the population that just simply don't like fish. And, of course, you've got vegans as well. So that's why omega-3 supplementation plays a critical role in increasing the omega-3 index in the larger portion of the population. So to answer your question, since everyone is uniquely different, testing is key. It allows you to understand your current status and, more importantly, take corrective action.
Actually, that's so true. And of course, knowing how we actually take the test and what to expect is crucial for those that are interested in taking the test to understand their omega-3 status in their body. Lalen, can you walk us through how these tests are done? What's the process? Where do you get the kits and so forth?
So the test is a dry blood spot test, which has actually been widely used by a majority of governments to do newborn screening, where they test for metabolic disorders or diseases. And more recently, it's also used to screen populations for HIV and hepatitis. So it's been around since the 1960s. The process is simple. You get a collection card, which is used to absorb a couple drops of blood via a lancet, and then you put that into a prepaid envelope and send that to the lab. And then once the analysis is done, you then receive the results. So it's an at-home test, which allows individuals to proactively monitor their health status or omega-3 status without the need of a traditional blood draw that you would typically do at a doctor's clinic. Furthermore, since the blood can be collected from the comfort of your home, the test is highly accessible, especially for individuals living in rural and remote areas who may not have easy access to a doctor's clinic or pathology lab. And it's affordable as well. Retailing for only $69.95 here in Australia, and it's available through various different retailers and websites.
And if I can jump in there, that's a really interesting point, and I'm probably showing my age here. When I initially went through training, we would do a blood draw just like Lalen's describing. So yeah, as a student, we would use a venipuncher, we'd draw out that blood, we'd take days to spin that down, ghost the membranes, analyze those membranes, and eventually come up with our fatty acid profile and our omega-3 index. And this has been a game changer in terms of being able to, exactly like Lalen's described, take research from what was very laboratory centric out into wider populations. And I think that's where we've seen the biggest difference in the last decade or two on this particular topic.
Yeah. How often should we be testing ourselves? Is it a weekly, monthly, quarterly?
We measure the fatty acids in red blood cell, and typically the life cycle there is around 3 to 4 months. So we would encourage people to consider retesting 3 to 4 months until you reach that desirable range. And then once you've reached that range, then you don't need to be testing so regularly.
Yeah, and look, from a research point of view, it's actually made such a difference for us as well, because if we want to carry out a trial that goes for months, we can actually, because it's fairly non-invasive and the burden on the participants less, you're not using a venipuncher, we can check more often. And that means we can actually observe that omega-3 not only going into the body, incorporating into the membrane, using the red blood cell as I think that's a really important part. We're using the red blood cell as a biomarker for all the other tissues. And that really gives us an insight to when peak levels for that particular dose has been achieved. So but part of what we can do now is we can look for when people have reached those peak levels, and we can keep them at those peak levels for as long as we want, and we can retest them. So it's been excellent from a research point of view as well.
And Greg, we talked about this in the first podcast series, that how long it takes to get to the right level, ideally over omega-3 index of 8, but it takes longer to come down. So that time of going from sort of low level to the omega-8 or above, what sort of time frame are we looking at, and how does, the level of omega-3?
Yeah, so that goes back into our research methods paper that we published last year, and many of the, particularly around the physical activity trials that we've worked in more often, many of them have been 4, 5, 6 weeks. Then we see some out at 8 weeks and some to 12. We really are looking, like Lalen's described, we are really looking at the lifespan or the turnover of the red blood cells. So we would advocate for omega-3 trials to be running for 12 weeks or longer as a minimum. And again, that's where the biomarker is invaluable, because we can follow that over not just the pre and post, but we can have a look at how quickly individuals are actually changing. And that gets back to that individual concepts that Lalen's described perfectly. Is it male? Is it female? What's the body mass? What's the physical activity? What's the gastrointestinal health of the person? These are all factors that could potentially influence the overall uptake.
And Lalen, just in terms of interpreting the results, what is a desirable range of omega-3 index?
So the report is designed for easy interpretation. The average consumer could understand this report. And essentially what we're using is a traffic light colour scheme on the report. So you have this chart, and then you have your results. Yeah. And then in the chart, if you're 0 to 4%, then you are highly deficient of omega-3s, and that would be the red zone. And then you've got the next criteria, which is 4 to 8%. So you're in the suboptimal range, so room for improvement. And then you've got the optimal range, which is between 8 and 12%, and that's in green.
It's helpful to have a clear benchmark for this aim, right? I mean, from a consumer perspective, who has interest in achieving that sort of level of omega-3 index.
And what's critical is that those have been born out of some really strong related studies that are related to cardiovascular disease outcomes and risk markers. But I think where the future is going is that potentially those traffic light systems potentially could start to be modified, because we could have other outcomes related to other physiological diseases or states. So we're really only just on the precipice of actually starting to use the omega-3 index in a range of different health and medical scenarios, I think is the best way of putting it.
Yeah, maybe just to add to that as well. I mean, in the contents of consumers or individuals wanting to do this self-care test where they, proactive about their health. So we try to design these reports in a way that it's really easy for them to digest the information, and also provide them with some tips on how they could improve their status through diet and supplementation. But then, of course, we have people like Greg who typically we would actually send the raw data of each of the individual fatty acids. But that segment, that consumer segment, is growing significantly. Consumers are becoming more savvy about nutrition in general, and they tend to be a little bit more proactive about nutrition and their well-being.
And it's an interesting point that it also breaks down a lot of the conversation about dietary fats and just lumping them all into one particular category. So now the consumer can actually break that diet apart and appreciate that not every fat is the same, and that there are very important roles for different fats like our omega-3 in the diet. So we don't just sort of put all fats into the same category that sometimes saturated fats and trans fats get lumped into. So it actually opens up positive conversations about the role of dietary fat in the human diet.
And that's fantastic insights. And that's a wrap for part one of our conversation on measuring omega-3s. Lalen and Greg, thank you again for the insights today, and we'll look forward to part two.
Thank you.
Thanks, Bassam.
DHA, the athlete's choice. Heart and brain, let's raise our voice. Muscle recovery will go far. With omega-3, your shining star.